But this is this deals with the rheology of the gel, how stiff it is. The stiffness is measured here on a logarithmic scale. This is time, and you can see this is stiffness 1 in terms of the units in which stiffness is, pascals. And originally nothing happens. This is the delay time. Let's follow the red line, which is elasticity. The blue line is viscosity, but they behave the same way. And then the red line goes up in a straight line. If you remember, a straight line on a semi-logarithmic plot is an exponential progress. So this progress is immensely rapid. Once it starts, it's almost explosive. And if you put it on an ordinary Cartesian scale, that is an x-y axis without the logarithmic thing, you can see that once this process starts, it takes off and goes on very rapidly, and you can imagine that would produce a great deal of pathology, which it does. So we thought, well, if we know something about the rheology, it would be useful to know something about the individual fiber. How stiff is the fiber? If the fiber is very soft, maybe the gel will be soft and won't create a lot of pathology. But if it's stiff, the opposite. So we looked at fibers, individual fibers in solution, and we measured how much they spontaneously fluctuate with Brownian motion. That's molecular hits induce the motion of a fiber. So over here is a flexible fiber. Over here, and you can see it moving this way, back and forth, fluctuating, and the magnitude of those fluctuations can be measured, and from that, with some not too difficult mathematics, you can calculate actually how stiff it is. If you do that with a stiff fiber, you'll see it fluctuates much less. So from that, we can get a measure of how stiff the material is, and it's very stiff. It's 1/10 as stiff as structural proteins such as actin and tubulin, but it's 1,000 times more stiff than proteins such as elastin, which are in the connective tissue and so forth, which is also a structural protein. So for a protein that's soluble, in principle, this is quite extraordinary that you can make such a stiff fiber. Well, this is the way we measured the previous thing. I won't delve into that because I made the point that you can measure stiffness with fluctuations. Then we also looked at how other structures can be made. It turns out that there are multiple fibers. You can have two fibers which stick together like this, which is shown in this video. I'm not going to get that video. Anyway, what happens is this fiber and this fiber just fluctuate around, and then they happen to join here for a moment. And once they join here, they're stuck together, they interact, and they just zipper up together till you just get one fiber here in the center. And the last thing that we did in terms of the mechanics of rheology is we managed to isolate a single fiber instead of a whole gel within a red cell. And we modulated it, that is to say, we let it grow, and you can see it deform the red cell like this. And then we plotted the amount of deformation on the x-axis as a function of time, and from that we can get a measure of how stiff the fiber is. So we can look at the stiffness, and also that relates to how elastic the membrane is. If it's very elastic, the fiber is going to go very slowly, and conversely. Okay. So the question is, what directions does the search for therapy go, or the search for ways to approach therapy, which is really the stage we're at? So we decided to look at what happens in the arterial circulation, the big arteries, like the brachial artery or the radial artery, in patients. Are there polymers? Did all the polymers dissolve in the lungs when they saw oxygen, or did some polymers come through? And you would imagine if some polymers came through, that could be potentially additionally pathogenic, because you wouldn't have to nucleate the next fiber, because you already had a nucleus from the residual stuff that didn't dissolve. So this is a patient. And the bright spots are red cells that have polymer in them. We looked at the birefringence. It's not important how. That's what we detected. And then we asked the patient to hyperventilate like that for 1 minute. And when he did that, we found a marked decrease in the number of cells that had residual polymers. In other words, we were getting his lungs to work more effectively. We were assisting breathing, and that assisted breathing managed to decrease the amount of polymers that failed to get dissolved. Then when the patient went to sleep, we see this increase in amount of polymer, and sickle cell patients very often have what's called sleep apnea or disordered breathing, which you would think would make the amount of residual polymerization greater, and this shows that it does. Well, this is just to show what birefringence. You have to know what's birefringent. If you have a polarizer up here and an analyzer down here, and you're putting light through, you get polarized light hitting the sample, and you analyze it coming out the bottom. And if you take a sample in between these two and you rotate it, it'll alternately, every 90 degrees, get light and dark. So we rotated this sample. This is one sickle cell. In this position, it's not very bright. Here's bright, and in this position, it's much brighter. So that confirms the brightness that we're seeing is due to birefringence, as it's called. But we also got an electron micrograph, and you can see all the fibers in this arterial red cell. So that confirms the fact that there are residual polymers, which was not known previously. So that's basically what I wanted to say. The avenue that we're pursuing is looking at the depolymerization end, but people have been pursuing many other avenues, and in particular, there's great activity in the area of adhesion of red cells to endothelium and adhesion of red cells to each other. It's a very active area. Hopefully, a useful cure or ways of treatment will arise from that. Thank you.